This is the third of 3 videos looking at the electron transport chain and oxidative phosphorylation. So you might want to go back and check out the other 2 videos if you haven't already. But with that caveat aside, let's just jump into this. What we're going to be asking today is how do mitochondria turn a pH gradient into ATP? So in the electron transport chain, which we've talked about a little bit thus far, electrons flow from NADH and succinate through this electron transport chain thing. They're being passed from complex to complex, from complex to mobile carrier, pumping, and in the process, we pump protons across the bilayer. What that does is generate a bunch of excess protons on this side, which then the ATP synthase complex can then capture to make ATP. And so what's happening in this video here is we're seeing protons flow across the bilayer from the IMS into the matrix. And then in the middle, what we're seeing is the formation of ATP. This is astonishing, actually. This is one of those molecular machines that the first time I heard about it, I was like, what? This cannot exist. It's literally a rotor that turns in response to a chemical gradient and then uses that to catalyze reactions. I mean, it is absolutely incredible. And what we're going to be doing is unpacking how this machine works in this video. Now, you might be looking at this and saying, well, Mike, what are you doing? You pasted in your slide your picture upside down. And it turns out I did. But what I'm trying to do is to be standard across all the things that I show here. I sort of pulled slides from different content, which I'll sort of cite as I go, but there's different standards for how people draw things. I am always going to draw this so the matrix is on the inside and the IMS is on the outside. And so we're going to be thinking about protons flowing this direction in all of the content that I show here today. Okay. So what are the parts of this ATP synthase complex? So I'm just going to label them here. We've got a rotor, and that's right here. This is part of the system that spins. We have a channel shown right here. That's 2. We have a stalk. That's this thing here that connects the rotor and the channel to the rest. We have a shaft shown here. And then finally, we have this thing called the AB trimer up here, this green thing. That is 5. And these 5 parts work together to allow this machine to extract energy or to make ATP from a pH gradient. So what does this look like? Well, here is another video of this. And what we're seeing here is this is a super schematized version of this. But hopefully what you can see is there's protons flowing this direction. There is some rotation happening. The red and the orange bits are rotating, and then the green channel stalk and AB trimer are staying the same. So what we see is the red part, the rotor and the shaft, they rotate, whereas the AB trimer is fixed. And I'm going to just duck for the second. Are fixed, that's what that says. The channel, the stalk, and the AB trimer are fixed. So part of it moves, part of it doesn't. So we're going to think about how this works by asking a series of questions. What causes the rotor and the shaft to rotate? And it turns out that the system is linking proton flow across the bilayer to rotation. So protons are coming in from, again, let's orient ourselves, from the IMS side, going into the matrix side. And what they are doing is as they flow downward, they are triggering a rotation of this complex in a circle. They're basically driving it in a circle. This is actually a really good view to pause on. If you look at this pause here, what they're doing is protons are coming in from the IMS side here. They are then rotating with the entire complex down here, and then they're being spit out there. So the protons themselves are actually spinning through the bilayer as they move through the system. And I guess just to make sure we're on the same page with this whole complex is embedded in a matrix, right? So we're seeing it spin. What we're doing is we're looking at if you sliced a matrix like sliced a membrane like an apple, and you're sort of looking down into the core along the edge. That's what we're looking at here with this sort of picture. Okay, so how does this lead to rotation? Well, it turns out that the protons bind at aspartic acids, sweep through the bilayer, and then exit at the exit channel. So right here, again, this is our IMS out here. This is our matrix. Again, I flipped it over to keep our sign convention correct. And what happens here is protons come in at this side, and then they exit at this side here. So they exit here, and they enter here. Okay? So that's the general flow. They do not go from here and then go that direction. Instead, they go this direction, sweeping all the way through the bilayer and then coming back on this side here. So they're driving rotation of this drum. Why? Why in the world does that happen? Like that actually, you think about it biochemically, that's very difficult to wrap your brain around why that's happening. Why do protons, so we're going to unpack that, we're going to ask 3 more questions. We're going to say, why do protons go from the IMS more often than from the matrix? How does a positively charged proton go through a hydrophobic bilayer? That's puzzling. And then why go all the way around? Why go this way and not just flip that way and dump the proton out? Okay, so let's tackle those 3 questions. So first, why do protons go from the IMS more often than from the matrix? And the reason for this is there's 2, actually. But if we think about it from just a concentration perspective, there are excess protons on this side, which means on average, you're going to see a proton want to move that direction more often than that side, just because there's more things on that side. If you've seen my analogy, or if you've seen my videos looking at flipping coins as a way to measure free energies, you have an excess of heads, right? So you're going to tend to see heads to tails transitions more often. You have an excess of protons on one side of the bilayer, so you're going to see protons moving across the bilayer from the IMS into the matrix more often just by chance. And so reason 1 is just a concentration difference. The other reason is actually has to do with the fact that these are positively charged, which means you have a net positive charge that builds up on this side of the bilayer, which means there's actually an electrostatic potential that's created. So there's energy that's being stored in the form of this charge gradient. And so relieving that charge gradient is energetically favorable. So there's also an electric potential that's set up. You could think about it, if you want to, in terms of the positive charges in the outside are attracted to negative charges in the inside. So from these 2 factors, from the concentration difference, which is encoded by this equation, and the potential difference, which is described by this, we can get the overall free energy. It says if we had to make a bet, we would see a proton going from the IMS to the matrix more often than we would see a proton going from the matrix to the IMS. All right, so that was the first question. That says why protons are going to want to flow this in one direction. Second, how does a positively charged proton go through a hydrophobic bilayer? Well, it turns out that these rotors have a bunch of negatively charged aspartates in hydrophobic pockets. And what that means is that a negatively charged residue, if this thing were to sweep to the left, a negatively charged residue would be stuck into a hydrophobic environment. That's energetically unfavorable because you're losing all of the contacts between that negatively charged thing and, say, water. And so energetically, that's not going to happen very often. But if you take a negatively charged aspartate and you put a positively charged proton with it, now you have a net neutral thing. So here, this would be a positive charge, neutral, positive charge, now neutral, positive charge, negative charge, now neutral, right? That means that neutral, these ones are neutral, and they can happily enter the bilayer. So an aspartate with a proton bound, so now it's the aspartic acid form, can enter the bilayer because it's neutral. The negatively charged ones can't. So that is how you take a positive charge and put it into a bilayer. You have a positive charge binds to a negative charge. So the offset of that negative and that, wow, that is terrible handwriting, even for my standards. So the negative and the positive together give you a neutral charge so that thing can enter the bilayer. Okay. Last question is why go to the left and not go to the right, right? So you can imagine a world where the proton comes in through here, and pops out through there. How do we avoid that happening? And it turns out it comes from this exact thing we were talking about with these negatively and positively charged residues. So at 1, what we have is a negative charge plus a positive charge. So that is neutral. So at 1, our aspartate residue is in the aspartic acid form. It is neutral. At 2, it is more negative. And the reason it's more negative is because protons are tending to fall off more often at 2 than they are at 3. So here at 3, more positive. So we've got excess protons here, and we've got less protons here. And so what does that mean for the system? What that means is this thing can ratchet to the left. It can ratchet that way, neutral, cool. It does not like going the other way. And the reason it doesn't like going the other way is think about what's happening back here at this position 2. That position 2 has dropped its proton off. It is now negatively charged. If this ratchets backwards, you're now taking a negative charge and putting it into the environment, right? So at 2, you have a negative charge that can go forward to 1, right? It can go to that more positive place, but it can't go backwards because there you're taking a negative charge and you're sticking it into the bilayer. So we flip to the left because we're taking neutral stuff. Neutral forward, awesome. Negative backward, not awesome. So that's where the switching comes from. What this actually does is it creates a ratchet that prevents reverse motion. So these guys are all in this neutral form, right? Back here, if it goes the other way, here it's in its negative form because the proton has fallen off. So it can't go backwards. It's like this ratchet here, right? It's spinning. It can spin in this direction, but it cannot spin in the other direction because if it did so, let's actually, guess we can't pause that video easily. It would actually sit there and stop it. It can't rotate in that direction. So we have a ratchet that's built around the charge state of the atom of these aspartates that net moves protons across the bilayer. And in case you're interested, we can actually see this experimentally. So this is a pretty nifty video. What these researchers did is they took a ATP synthase complex, they mounted it on a surface, and then they attached an actin filament, which is a long linear filament of proteins that they can label with a fluorescent dye. And then they just used something called turf microscopy to watch the motion of this. And hopefully what you can appreciate is this thing is spinning around. That's that long filament that's spinning around, being driven by ATP driving this complex forward. So this complex really is spinning, and as it's spinning, it's moving protons across a bilayer. Okay. So we've gotten to the point now where hopefully you understand why protons are wanting to move this direction. It's because they have an excess concentration. Concentration gradient makes them want to flow from the IMS to the matrix. And hopefully you have got a better understanding now of why you get rotation, that electrons can flow, or protons, sorry, flow in this direction in the neutral form because the negative and the positive interact. And then it can't go backwards because that would now be taking a negatively charged thing and putting it back into the bilayer. Last big question is how in the world can you use rotation like this to actually do a reaction like the synthesis of ATP? And so to look at that, we're going to recall something that maybe you've seen before. I've talked about it in some content, but maybe you've encountered it in a different class or the like, and that is hemoglobin. Hemoglobin is this oxygen-carrying molecule that fluctuates between an R state and a T state. This is a set of videos made by Janet Owasso where she showed different forms of this. You can see it opening and closing, opening and closing. And the idea is these move cooperatively. The protein is either all in the R state or in the T state. It fluctuates between them. It doesn't hang out with half of it being R and half of it being T. Okay. If you've never heard of hemoglobin, don't worry about it. Hopefully that was helpful if you have heard of hemoglobin. So now let's look at this one. And it turns out this one has cooperativity, but it's got cooperativity that's quite exotic. Instead of having two states that's fluctuating between R and T, where everything is either R or T, it has three states, L, T, and O, and it cooperatively goes between them where it has to be one is in L, one is in T, and one is in O. So as the rotor rotates, it's spinning this shaft, which is driving this whole system cooperatively between these states, L, T, and O. Let's look at this in what we're going to do is look at it in a sort of movie form, and then a super schematized form, and then back in the movie form. And so this is this animation made by the MRC, the Medical Research College. What they're showing is the system going forward. Hopefully what you can see is this shaft in the middle is rotating. It's spinning through a set of states. And then these molecules, the yellow and orange ones, are transitioning between functional forms. They are this AB trimer. And what you hopefully appreciate is now they're in one form. As it rotates, the shaft rotates, they go into a different form. As the shaft rotates again, they go into yet another form, and so on. As it continues to rotate, it's driving these yellow and orange molecules through a set of different conformations. Okay, whoops. So now let's look at this in super schematicized form. This right here is that shaft that's sticking down from the channel. Sorry, this might be helpful to actually look at what I'm trying to gesticulate on. It's sticking down from the channel. This thing is spinning. It's being spun by the ATP or the protons flowing through the gradient. And then the trimers, the alpha beta trimer part, is going to have one subunit in O, one subunit in T, and one subunit in L. So hemoglobin, remember it's all R or all T. This is one must be O, one must be L, and one must be T. And this is enforced by the chemical interactions between these two, right? The sort of nature of the molecular surface. And O on its bottom side is going to want to interact with a T, and a T on its top side is going to want to interact with an L. So these interfaces, molecular interfaces, actually enforce this OLT rule. Whichever way the shaft is pointing is going to be in the O conformation. So this will be O, which will enforce this being T, which will enforce this being L. All right, we are getting there. Now the shaft is going to actually rotate in a counterclockwise direction. So as protons flow through the gradient, as they flow through that, or flow along the gradient as they flow through that channel, it drives rotation of this shaft, and that shaft is rotating between states. Remember, the AB trimer is not rotating, just the shaft. So what we're seeing in this little mini animation is anywhere that the shaft is pointing is the O form. It's stabilizing that form. And so as the shaft rotates, it's driving different proteins through each conformation. So let's actually look at a single subunit, and then we'll go back to the whole subunit. So what I'm showing here is a single subunit going from O, shaft rotates, now that's in L, shaft rotates, now it's in T. Points back where in O goes L and T. There's a stereotypical order that's happening where this molecule is being driven between the O form, the L form, and the T form by the rotation of that shaft. Okay, what does that help with? Sorry, had a problem. What that helps with is it drives a catalytic cycle. So that's because these different forms differ in their affinity for ADP, inorganic phosphate, and ATP. So the L form binds to ADP and PI, and I'm drawing that very schematically as it binds to this pentagon and this triangle. The T form, and I'm going to disappear here, the T form brings together the ADP and inorganic phosphate, so those now interact with one another. They're sort of going from being in this pulled apart form to this close together form. Another way to put that is that this state has a high affinity for ATP. And then the O form actually doesn't bind to ATP, ADP, or inorganic phosphate well at all. So what does that mean if we stitch it together? What that means is that this cycle catalyzes production of ATP. So when this molecule is in the O form, nothing binds at all. When it's in the L form, inorganic phosphate and ADP will bind. The shaft drives this whole system. Now it goes into the T form and forms ATP. It rotates again. Now you get the O form and the ATP falls off. So as the shaft rotates between states that have different affinities for ADP, for inorganic phosphate, and for ATP, it catalyzes the production of ATP from ADP and inorganic phosphate. Super, super cool. And this is actually happening on 3 subunits at once. So remember, the subunit itself is not rotating. The subunits are fixed, but the shaft rotates, and whichever it's pointing to is stabilizing that form. But since there's this block of 3 subunits, you always have an O, you always have an L, and you always have a T. But each one of them is being driven through this cycle. So let's look at this, let's say the top left, L, T, O, right? It's going through this conformational change, but that means every spin gives 3 ATPs. So you do 3 turns of this, one full turn generates 3 ATPs, 1 at each of the 3 binding sites in the AB trimer. Okay, right, so rotation of the shaft is pushing this trimer through LTO, which is catalyzing the formation of ATP. I said I'd go back to the main video. I'm going to go back. Sorry, I meant to put in a slide for that and I failed, but that's okay because I can go backwards. So now let's look at this the zoomed out form of this now that we've seen the thing in a schematized form. So in blue, we have the shaft. In the bottom, we have these AB trimers. And now you can see it going through the different states as it binds to ATP and catalyzes its production. So let's scrub backwards here. This form that we're in is the stop. Right here, we are now in the L form. We've bound to ADP and inorganic phosphate. The shaft rotates. We are now in the T form. We formed ATP by driving it into the T form. And then if we keep going, eventually it rotates one more time. And now we are in the O form where that ATP no longer binds, right? So by this shaft rotating, driving these conformational changes, driving the protein from being in the L form here, binding to the subunits, the T form, which is going to catalyze the formation of the ATP, and then the O form, which is going to catalyze the release of the ATP, we end up with a full catalytic cycle. Okay, so that is what I wanted to go over on this. Really quickly, I just wanted to point out that overall, this yields a ton more energy than glycolysis alone. So we get 2.5 ATPs for every NADH. We get 1.5 ATPs for every succinate. It's probably a better way to count it. And these actually aren't integers because this isn't a simple chemical reaction, right? We've got a proton flow that's coupled to a phosphorylation reaction, but still you're getting a lot of ATP molecules out. And if you go through and add up all of the different parts of this from the citric acid cycle and glycolysis and everything, what you get is that you get 32 ATPs per glucose out of this process of the electron transport chain plus oxidative phosphorylation. If you contrast that with glycolysis, we got 2 ATPs per glucose. So you get significantly higher energy yield by doing all of this amazing complexity. So, okay, well, hopefully that's given you a taste for how this cool molecular machine works, this ATP synthase, harnessing proton flow to make ATP. Look for a couple videos in the future as I go through it. I'm going to unpack some of these mechanisms in more detail. I skipped over, say, complex 1 and complex 3. There's some really cool things that happen there that I'll talk about mechanistically. But for now, hopefully you have this picture of what the electron transport chain and oxidative phosphorylation are. So with that, I will leave it here, and thanks for watching.